Hi, this is Dr. James, and I'm going to go over with you our first lectures, just in case I'm not here with a new grandbaby, but also to give you something to go back and listen to over and over again. I'll be talking about content from Chapter 7 and Chapter 8, just getting you started about conception, fetal development, fetal circulation. These are some of those things that you really need to know about. I'm not going to test you real heavily on it, but if you don't have a good understanding, it's very hard to continue to develop in that area. And I'd like to welcome you to Nursing 2044, talk a lot about moms and babies, and it's usually a really, really fun course. I will try to give you some pre-work each week to kind of get yourself acclimated to what we'll be talking about. I like to use a lot of active learning. So one of the things we'll also be doing is taking a little assessment of how we actually learn best, so that I can make sure to have activities that appeal to those of you who can't be still, and those of you who are very visual, and those of you who like to read and study. So I'm going to try to get myself out of the way. I'm going to share my screen with you. So bear with me. All right, and hopefully you're able to see my PowerPoint so that we can continue on. And if you see me looking away, that's because I'm looking at what I'm presenting to you. This is maternal newborn nursing. We know that families come in all shapes and sizes, traditional and non-traditional, and we'll be talking a lot about that. And we'll also be talking a lot about how culture plays an important role, and how we as nurses want to make sure that we support all of our families, no matter what culture, ethnic background, or where they come from. So I always like to start with this little note from Dr. Seuss, and he explains pregnancy. What is a womb? Well, a room is a room. A womb is a room where a kid will be soon. A womb is all squishy, all moist and magenta. And if there's a kid, then it's full of placenta. But the kid must be small, if there's one there at all, just the size of a bean or the size of a ball. When the kid gets too big, then it's time to get out. When it happens to you, you will probably shout. You'll shout, you'll yell, you may roll on the floor, because the kid must come out of a very small door. And we'll be learning a lot about how children are developed and how they're born. And guys, sorry, but you got to live through it this time. You'll have this in my PowerPoints. It's a hyperlink to a video. I won't take the time now to show it to you. It's birth in 4 minutes. Some of you have probably never even seen a delivery. So this is a very good video that'll take you through the whole thing. This is our textbook, Maternal Child Nursing Care. It's published by Pearson, and we're going to talk a little bit about conception and fetal development. So a little bit of patho, A&P, you guys should, this should just be a review for you, so I'm not going to spend a lot of time on it. If you don't understand what the difference between mitosis and meiosis is, then review it in your patho or your A&P book. We know that mitosis is cellular division. The daughter cells are exact copies of the original cell. These are identical to the parent and to each other, and these cells contain a full set of chromosomes. So these are the process is the cells are referred to as diploid, and the somatic cells will continue to reproduce and replace each other. So the cells will undergo several changes during cell division, and at the last phase, the cell cytoplasm is developed, and the parent cells is going to divide again into two daughter cells, and each of these has its own nucleus, and these are identical to the parent. And to end cell division, they each have the same diploid number of chromosomes, 46, with the same genetic makeup as the cell from which they came. So it's all identical, all part of cell division. Meiosis is cell division that produces the reproductive cells, which are called gametes. These are the sperm and the ova that are necessary for reproduction. And each cell has half of the genetic material of the parent cell. So whenever you're a young lady, your ova has half of the genetic material from your parent, and the sperm has half the genetic material from the parent diploid cells. Two successive cell divisions happen in meiosis. The first division is whenever the chromosomes will replicate, and the second division is when the chromatids from each chromosome will separate and will move to opposite poles. And then division occurs and 4 cells are formed. There are 2 successive cell divisions in meiosis. There's a haploid number of chromosomes, and the daughter cells only contain half of the DNA of the somatic cells. So I'm going to give you a quick and easy table that helps explain both of these processes, because as you can tell, it can get very confusing. This is a table that is in your textbook. Mitosis, it gives you what the purpose is, and what happens is just one stage of cell division where 2 daughter cells are identical and we have 46 chromosomes. And then when meiosis occurs, this is where our reproductive cells are formed. There's a reduction in the chromosomes by half to a haploid, which is 23 chromosomes. So whenever fertilization happens, then the diploid develops again. So you can read that, go over it. Am I going to test you heavily? No, this is just something you need to know and be aware of how it happens. So now we're going to get into reproduction, oogenesis. This produces the female gamete called the egg or the ovum, and it begins to develop early in the fetal life of a female. We're born with the number of eggs we're going to have for the rest of our life. The ovaries begin to develop early in the life of a female, and we have all that we're going to have whenever we're born. During puberty, the mature primary oocytes continues through the first meiotic division in the ovary, and the haploid cells are released during ovulation. And when we ovulate, if there's not a sperm there to fertilize, then a diploid is not formed. During the first meiotic division, 2 cells of unequal size are produced with the same number of chromosomes. One cell is secondary and the other is the minute polar body. And you can read all of this. Each contains 22 double-structured autosomal chromosomes and 1 double-structured sex chromosome, the X chromosome. And this is where genetics comes into play, and we do not go in depth in genetics in this class. And this is just is a reinforcement of what oogenesis is and the first meiotic division. So the sperm, the production of the male gamete occurs during puberty. The primordial germ cell begins with a complete set of genetic material, and here again, when it finishes, then we'll have a diploid number of chromosomes, the full set. The cell replicates before it enters in the first meiotic division, and now we have a primary spermatocyte. And the primordial germ cell, during the second division, it will divide and form 4 spermatids, each with a haploid, and after it is complete, whenever it meets an ova, then it will have a diploid. And you can read all this. The male is a Y sex chromosome. And here's a chart you can look at of what the result is for a male and a female. So fertilization, most of us realize an ova and a sperm have to get together. The preparation is the first component. An ovum is released in the fallopian tube, and this is during ovulation. It is viable for 24 hours. The sperm must be deposited in the vagina, and a sperm can be viable for up to 72 hours, but it's most highly viable during the 24 hours. And a sperm and an ovum must meet each other to become fertilized. So the sperm penetration will cause a chemical reaction, and it will block more sperm from coming in. The sperm will enter the ovum. There's a chemical signal that prompts the oocyte to complete its second division, and true moment of fertilization will occur whenever the nuclei of the sperm and the ovum unite. The chromosomes will pair up to produce a diploid, as we talked. Each nucleus has 23 chromosomes, and the union will create the diploid number of chromosomes, which is 46. And so now the new little zygote will contain a new combination of genetic material from the mother and the father. The sex of the zygote is going to be determined at the moment of fertilization. 2 chromosomes of the 23rd pair is the sex chromosome. It'll either be a double X or XY, and this will determine what the sex of the individual is. Females will have 2 X's. A male will have an X and a Y. So here's a little image of how the sperm will penetrate the ovum and the sequential steps of penetration by a sperm. And you can look at this, and it's also a picture in your book of how sperm get into the ovum. It's a very quick, distinct process, and it's amazing how the human body has developed and how this process actually works. Here's another picture of a sperm penetration of the ovum. You can see there's lots of sperm swimming around all over the place, but all it takes is one. The changes to the ovum once it is fertilized from conception to implantation. This gives you a visual of the ovum from the fallopian tube, and then where it has to go to penetrate into the inside of the uterus. You'll see the inside of the uterus with where it says implantation of the zygote. And you see where fertilization takes place inside the fallopian tube. And the key is that the ovum continues to travel into the appropriate area and not implant inside the fallopian tube. So once it happens, once fertilization happens, there is a rapid cell multiplication. And you can read all this. We're not going to go into all the detail, but you do need to know implantation does occur within the next 7 to 10 days after it's fertilized. After 10 to 14 days, the ectoderm, mesoderm, and the endoderm. This is where all the tissues, organs, and the organ systems develop of the fetus. More cell division goes on, and the organs and the organ systems develop from just these small primary germ cell layers, and it occurs in 10 to 14 days after fertilization. The embryonic membranes, the chorion and the amnion, those form at the point of implantation. And here's a really good table, it's also in your book, of the different body structures that are formed from the different layers, from the ectoderm, mesoderm, and the endoderm. This is for your knowledge so that you'll have a good understanding of what is formed in those first few days. It's very interesting. The amniotic fluid is created when the amnion and the chorion grow and connect, and this is what forms the amniotic sac that protects the fetus for, some people like to call it 9 months, we call it 40 weeks until the baby is born. Also, the yolk sac develops. This produces primitive red blood cells. These will be incorporated into the umbilical cord as the baby continues to develop. All right, here's another good image for you of how the primary germ layers are formed. This is an implantation of a 7.5-day blastocyst where the cells of the disc are separated from the amnion. And then the picture B is at 9 days, and C shows you even further development and division by day 16. So we've talked about the 3 divisions. The differentiation of the endoderm is the epithelial lining that is there for the respiratory system and for the digestive tract. And we've already established all the tissues and the organs are established from these germ cell layers. So here's a 5-week embryo. You can already make the distinction between the mouth, the eyes, the nose, where the pharynx should be located, the lungs, you see the big liver there in the center, and of course the umbilical cord. All the major organs and organ systems have been developed. Okay, we're going to talk a little bit about how twins are formed. This is a formation of fraternal twins, and you notice there are 2 separate placentas, 2 different sacs. They fraternal twins are from 2 blastocysts, 2 separate individuals, even though they are implanted and born at the same time. They are developed by 2 ova and 2 sperm, not just 1. Now, identical twins are born from 1 ovum and 1 sperm, and you see that they share 1 placenta. They just have 2 amnions. If you see that, there are 2 sacs. They're developed from 1 blastocyst, and this is why they're considered identical. So, monozygotic. This is a term you might need to remember. Monozygotic twins are developed from a single fertilized ovum. They are of the same sex and same genotype. Identical twins have a common placenta. Monozygotic twins is not affected by the environment, race, physical characteristics, or fertility. Both fetus are the same. sex, same characteristics, and single placenta. That's a good way to remember monozygotic. One placenta, same sex, same characteristics. Usually the division happens within 4 days of fertilization. You have 2 embryos, 2 amnions, 2 chorions, and usually you're going to have monozygotic. We're not going to get real deep into twins because really we could talk about it all day. They have 2 separate sacs, and these sacs will eventually be covered by 1 chorion, which is attached to the inner lining of the placenta. You can just read the rest of this detail about monozygotic twins. Dizygotic twins. There are 2 separate ova fertilized by 2 separate sperm, 2 placentas, 2 chorions, 2 amnions. Sometimes the placentas might fuse, making it appear as 1, but there are still 2. These are fraternal twins. They oftentimes have no similarities to one another, and they're usually no different than siblings born at different times. They can be the same or different sex. So we're going to talk about further more of the development from reproduction to help you know what's happening. The umbilical cord, this is developed from the amnion. This is what connects the baby to the mom, and no, mom's umbilicus is not connected to the baby's umbilicus. The umbilicus is where we are cut from when we're born from the mom. The umbilicus connects the baby to the placenta. This is a stalk that attaches the embryo to the yolk sac, and it fuses with the embryonic portion of the placenta, and this is the pathway from the chorionic villi, which is inside the chorion, which is the lining of the placenta, to the embryo. The umbilical cord has 2 arteries and 1 vein. It's protected by a jelly-like substance. It's called Wharton's jelly, and this protects the vessels because they are very, very delicate. They're very, very small. The function of the umbilical cord is very important. This is a circulatory pathway to the embryo. This is how the embryo gets its nutrients. This is how the embryo gets its oxygenation. And this is how the embryo gets rid of its waste. The placenta begins by the 3rd week of embryonic development, and it develops wherever the embryo attaches to the uterine wall. So sometimes it can be attached down low close to the cervix, and sometimes it can be attached on the side of the uterus. Sometimes it can be not attached very well at all. The function of the placenta is for metabolic and nutrient exchange between the embryo and the maternal circulation. It has 2 parts. It has a maternal side and it has a fetal side. The fetal side is very shiny and smooth. The maternal side is very rough because it's more vascular, attached to the mother side. On the maternal side, like I said, it's very vascular and it's red and flesh-like. You might hear some people, whenever you go to the hospital, some of the nurses may be calling it a dirty Duncan, but the reason it's red and fleshy looking is because it is very vascular, because it's attached on the inside of the mother. The baby side is the chorionic villi and their circulation, and it's covered by amnion, so it's very shiny and gray. So you may hear nurses or healthcare providers calling it the shiny Schultz. Here's a picture of the maternal side. As you can see, it's pretty rugged looking. And here's what the baby side looks like. So it's kind of silvery, kind of shiny. It's smooth, so that's why we call it the shiny Schultz. All right, we're going to talk about growth and development a little bit and what happens. And there's some very good charts in your book, so it would behoove you to read over it and have a good idea of what's going on. So this is what an embryo looks like at 5 weeks. It's more of a C-shaped body, and it does have a tail because this is early development. And with our technology now, even though the embryo is at 5 weeks, if we use a transvaginal ultrasound, a regular ultrasound, you may not be able to pick it up, but a transvaginal ultrasound would be able to pick up a heartbeat as early as 5 weeks. So at 4 weeks, the fetus is 4 to 6 millimeters in length. The brain is formed. The limb buds, you can see a heartbeat, and the GI system begins. And at 6 weeks, the fetus is 12 millimeters in length. It has a very primitive skeletal shape. You're able to visualize the chambers of the heart. Of course, this is with not regular ultrasound. This is with more internal. Respiratory system begins and the ear formation begins. This is to let you know how early all of these things begin to occur. At 12 weeks, the fetus is 8 centimeters. The skeleton begins to ossify. The liver produces red blood cells. The palate in the mouth is complete. The skin is pink. Thyroid hormone is now present. Insulin is also present in the pancreas. Now, is it mature? No, it's still immature. At 16 weeks, the baby is 13.5 centimeters. The teeth begin to form. Meconium begins to collect in the intestines. The kidneys will assume shape, and the fetus begins to develop hair on the scalp. So here's a picture of a fetus at 20 weeks gestation. You'll hear me talk often in class. 20 weeks is the age of viability. A fetus who is younger than 20 weeks often will not be able to live outside of the uterus, but once they're at least 20 weeks, some fetuses and babies are able to live outside the womb. At 20 weeks, the fetus weighs about 435 to 465 grams and will measure about 7.5 inches. They'll have brown fat on the skin. The skin won't be as transparent. Like earlier than 20 weeks, the skin is very translucent. You might be able to see some woolly hair over the head. You'll be able to see fingernails over the fingers and the toes. At 20 weeks, 19 centimeters long, myelination of the spinal cord starts. The baby starts to suck and swallow. Lanugo is what the woolly hair is over the top of the head. And it also covers the whole body because this is what helps keep the fetus warm. And then there's a cheesy substance. It's called Vernix caseoso. And that is also all over the body to help protect the body because the fetus is inside a bag of water. And it could really make the skin peely if it were not for the Lanugo and the Vernix. Surfactant production begins, and that helps with lung development. And the brain begins to appear mature at 20 weeks. And we are talking about normal fetal growth and development for several weeks. And I will let you know whenever we're starting to talk about the abnormals. At 28 weeks, baby's 27 centimeters long. The nervous system starts to regulate some of the nervous system functions. The fetus has adipose tissue, nails, eyebrows, eyelids, and now the eyes are open. And at 36 weeks, the baby is 35 centimeters long. You have the earlobes are soft with very little cartilage left, and you're able to start seeing the creases in the soles of the feet and in the palms of the hands. At 40 weeks, Between 38 to 40 weeks, the baby is considered term. The baby should be 40 centimeters long. There's adequate surfactant in the lungs. There's vernix still left in the skin folds, and the infant will only have, and I keep saying fetus, infant, baby simultaneously. It's still a fetus until it's born. Lanugo on the shoulders, the earlobes are very firm now, and we can tell what sex the fetus is. Usually by now, fetus weigh between 6 pounds 10 ounces to 7 pounds 15 ounces. Now we're in the healthcare facility. We measure this by grams, so this 3,000 to 3,600 grams. But that weight varies dependent on what ethnic group they are from. The skin is very smooth, it's polished. Head on its hair is usually very coarse and about 1 inch long, and the body and extremities are usually pretty plump. So here's a really good chart that is good to give to our clients to help them know what's happening when, what they really need to know. And it just kind of lets them know, well, whenever you're about 16 weeks pregnant, this is what's going on. It's also very helpful to us. It helps us to have an idea if we don't have children, if we haven't experienced that with our families, to kind of know what's happening in each week. Okay, so we're going to talk just very briefly about things that might influence congenital malformations, and that'll be it for Chapter 7. All right, things that can adversely affect embryo development are teratogens, and this is anything that the mother may be exposed to within the first 8 weeks of cellular division, and it can adversely affect organ development and fetal development. And what I'm talking about are drugs, viruses that she may be exposed to, or any type of radiation that she may be exposed to. And this depends on the maternal and fetal genotype, the stage in development, when the exposure occurs, and what the dose and duration of exposure is. So that's why a lot of our drugs that we give, their pregnancy categories attached to them. And whenever you're working on your drugs for this course, I'll want to know what's the pregnancy category about that drug. And you'll have experience with some mothers who are addicted or have been addicted, and we'll talk more about those things. But realizing some of these things that they consume are painkillers or anything, and she becomes pregnant and they are teratogenic to the fetus, she may end up losing the fetus. But that's really all we're going to talk about with abnormal congenital development for the first few weeks. All right, that's it for Chapter 7, and I'll be back with you. We're going to continue on with Chapter 8.